In 2017, UC Davis and the Yurok Tribe Environmental Program received funding from the National Superfund Research Program for a collaborative project to address concerns with contaminants in the Klamath River. These contaminants may come from multiple sources, including illegal marijuana cultivation, the legacy of timber harvesting and processing, and agricultural runoff from the upper watershed. In this video, you will hear about Project 3 of the UC Davis Yurok Tribe Environmental Program Superfund project, which focuses on developing techniques called immunoassays that allow us to track and measure the impacts of exposures. Immunoassays are tests used to detect the presence or quantity of a substance that may cause an immune response. You will hear from Dr. Natalia on her work to measure when toxins produced by blue-green algae called microcystin are present in the environment. Eventually, Natalia's project and Tom's measurements will connect to a third project to develop a lab-on-a-chip handheld device that Yurok community members can use to determine whether toxins are present in the water.
So hello, my name is Natalia and I'm project scientist. So I'm a part of the project that develops assays for monitoring and quantification of environmental contaminants. I oversee the overall progress of the project, but also actively involved in the development of these assays. Microcystin is a toxin produced by blue-green algae, and then development and proliferation of algae blooms most probably result from combination of environmental factors as availability of the nutrition, temperature, sunlight, but also water chemistry. And as for now, it is unsure what is specific cause of algae blooms, but industrial contamination might be one of those factors. Classical immunoassay is performed using this plate that contains 96 wells. So each well represents a reaction or a test. So we will be running 96 reactions in parallel. And some of these wells will be used for a calibration curve that uses known concentration of microcystin. And the rest of the wells will be used for the samples collected from different sites on Yurok territory. And then the operator will follow the protocol where different reagents added in a certain sequence, and at the end we will observe development of color, and then this color we can compare with the calibration curve and then correlate the color with the concentration of microcystin. And the assay could be used by community members when they have a concern about water quality or on a regular basis. We are currently establishing microcystin immunoassay in our lab, and once the assay is ready, we will train Yurok Tribe scientists to perform the assay. And this means that Yurok community will have a rapid test for analysis of level of contamination in the river, and that will inform them as to whether it is safe or not to have any water-related activities. And I know that there is a microcystin monitoring program established, but they receive samples 1 week or 2 weeks later after sample collection. So immunoassay analysis will provide people with more relevant and timely information.